Hi, my name is Soumya Pillai. Welcome to another episode of Pure Science. To set the context of this week's science breakthrough, what if I told you that some people in their 80s have the memory of someone in their 40s or 50s and that their brains actually look younger at the cellular level? Scientists call these individuals superagers. These are people over 80 years of age, but when tested, they perform on memory exams just as well as or even better than people decades younger. For years, researchers have wondered what makes their brains so resilient, but a new study from Northwestern University, Feinberg School of Medicine, now offers one of the clearest biological explanations yet. The findings show that superagers don't just act younger cognitively, but parts of their brains actually retain youthful molecular features. To understand this, we need to start with a small but critical structure in the brain called the hippocampus. This region is essential for forming new memories and learning new information. It is also one of the first areas affected in age-related cognitive decline and in Alzheimer's disease. As we grow older, the hippocampus typically shrinks. Connections between neurons weaken, and one process in particular slows down dramatically. This process is called neurogenesis, which involves the creation of new neurons. For a long time, scientists debated whether older adults could even make new brain cells at all. This new research adds powerful evidence that they can, but not everyone does it equally well. The researchers examined donated brain tissues from several groups, including young adults, typical older adults, people with mild cognitive impairment, individuals with Alzheimer's disease, and superagers over 80 who had exceptional memory while alive. But instead of simply looking at the brain under a microscope, the team used advanced molecular tools to study how genes are regulated inside individual brain cells. This is where the story gets interesting. Inside every cell, DNA is wrapped around proteins in a structure called chromatin. Think of chromatin as packaging. If it is tightly packed, certain genes are hard to access and remain switched off. If it is loosely packed or open, so to speak, genes are easier to activate. This openness is called chromatin accessibility, and it matters because it determines which genes a cell can use and therefore how that cell behaves. What the researchers found striking was that in superagers, certain brain cells had chromatin patterns that looked much more like those of young adults than those of typical elderly individuals. In other words, at a molecular level, parts of their brains had maintained a more youthful state. The biggest difference appeared in cells involved in neurogenesis. Let me tell you how. Neurogenesis begins with neural stem cells. These are early stage cells that have the potential to become neurons. They then develop into neuroblasts, which are cells that are on their way to becoming neurons, and eventually into immature neurons that later mature fully and integrate into brain circuits. In typical aging, many neural stem cells are still present, but the process seems to stall. Fewer of them successfully mature into functional neurons. In Alzheimer's disease, this slowdown appears even more pronounced. But in superagers, the pathway looks different. The researchers found significantly more neuroblasts and immature neurons in the hippocampus compared to typical older adults. That suggests that in these individuals, the brain continues to produce and develop new neurons even in their 80s. It is not just that they have stem cells, it is that those cells are successfully moving through the full developmental process. And that could be crucial. New neurons in the hippocampus are thought to support learning, flexibility, memory formation, and the ability to distinguish between similar experiences. The researchers also identified what they call a cognitive resilience signature. This is a distinct pattern of gene regulation seen through chromatin accessibility that was shared by young adults and superagers but reduced in typical aging and disrupted further in Alzheimer's disease. Thousands of regions in the genome showed differences in accessibility, particularly in cells tied to neurogenesis and synaptic communication. Now let's talk about synapses, which are the connections between neurons. Memory doesn't just depend on having neurons. It depends on how well they communicate. Excitatory synapses, which helps neurons send activating signals to each other, appeared better preserved in superagers. Support cells in the brain, such as the astrocytes, also exhibited regulatory patterns associated with healthier aging. So, what does this mean? First, it challenges the idea that cognitive decline is inevitable. Aging does affect the brain, but this study shows that some individuals maintain molecular programs that support continued neuron development and stronger neural connections. Second, it suggests that resilience in aging isn't just about avoiding damage. It may also be about preserving youthful gene regulation patterns, keeping the biological machinery that supports neuron growth and communication active for longer. There are still limitations. The number of superager brain samples available for study is small. And this type of research looks at brain tissue at the end of life, giving us a snapshot rather than a movie of how these changes unfold over decades. If scientists can understand what allows superagers to maintain these youthful chromatin patterns and continued neurogenesis, future therapies might aim to replicate those mechanisms. Instead of focusing solely on stopping degeneration, researchers could work to actively support brain renewal. In simpler terms, this study suggests that some 80-year-old brains aren't just surviving, they are still renewing themselves. They retain gene activity patterns that resemble younger brains, continue producing new neurons more effectively, and maintain stronger cellular support systems. But it is important to clarify that the researchers did not investigate lifestyle, diet, education, exercise, or genetics to explain why someone becomes a superager. They also do not claim that making more new neurons directly causes exceptional memory. What the study identifies are biological signatures, measurable differences inside the brain that are associated with cognitive resilience. In other words, this research shows what is different about the superager brain, but it does not yet explain what leads to a person to develop that kind of brain in the first place. But we do get a sense of how aging is not a single fixed pathway. The brain's trajectory in the later life can vary dramatically, and in some people, it remains remarkably resilient. That was all from me this week. I am Soumya Pillai, and you were watching Pure Science.